7 years or so, rising through the ranks. She did her residency and postdoc training at Johns Hopkins University. And then even before that, she was a MD PhD student at Baylor College of Medicine with Marty Matcha. At Johns Hopkins, she really built this amazing program to study transposons, which typically, like myself, I study transposons in a very basic science setting using model organisms like fruit flies and cells. And so most of the studies with transposons have generally been very fundamental and basic, but Dr. Burns' lab managed to bring transposons more to the forefront in a translational manner. She developed this fantastic antibody to the line 1 open reading frame protein 1 that has been really a game changer in detecting the expression of transposons in cancers and tumors, and has really kind of brought this kind of somewhat esoteric or genomically very challenging subject of transposons because they're so repetitive. She's kind of brought it to the forefront that it can actually be studied as an agent of human disease. Her CV is like 43 pages long, which I'm not going to read everything about that, but she's done a lot of work in the transposon field as well as in the mentoring arena, mentoring a lot of MD, PhD students, fellows, postdocs. She's really kind of rising the ranks quickly in the genomics and human genetics field. She's organized many conferences on transposons. In fact, I was supposed to meet with Kathleen at the Keystone Conference this February that got unfortunately postponed. Hopefully it will still happen, but it got postponed because of the pandemic. But luckily we were able to reschedule her visit here today. So I'm really looking forward to her talk about transposons and cancer. And one last little plug, she just published a single author perspective in Science on repetitive DNA and disease. So if you miss anything from this talk, check out her perspective in Science as another plug for her great work. Thank you for coming to BU, Kathleen.
Thank you so much for the invitation. And this is really just terrific to, after 2 years, get out a little bit in my new town and meet faculty and students at the BU. I'm really looking forward to the day, and I'm very grateful for the invitation. Hope it's the first of many conversations. So I'll be speaking primarily about our work on transposable elements in cancer. This audience may need very little introduction to the fact that transposable elements have had a huge role in shaping our genome. So like all other complex eukaryotic genomes, much of our inherited genetic sequence is interspersed repeats derived from the activity of mobile genetic elements. Once I understood a little bit about that biology as a graduate student, I became fascinated by these sequences and wanted to better understand how they might relate to human disease. And so my story sort of begins in thinking about them as agents of genetic variation, and then more recently we're thinking a lot about their expression and activity in malignancies. All in, about half of our DNA is immediately recognizable as belonging to one of these transposable element families. About 20% of our genome are so-called short interspersed elements. I'll talk about them a bit as genetic variants early in this seminar. About 20% of our DNA are the so-called long interspersed elements. These are protein coding, still active mobile genetic elements in humans today, and we're very interested in the expression of line in cancer. I'll spend really most of our time together focused on that biology. A relatively smaller slice of our genetic makeup is owed to the activity of endogenous retroviruses. They're dead in modern humans as mobile genetic elements, although they've been recently active, and so continue to be a source of genetic polymorphisms inherited in human populations. And many endogenous retroviruses still retain the potential to be expressed and potentially have gene regulatory effects or generate RNAs or even residual protein coding units that may be important to understand in human development and disease. And then a very small amount of our genetic makeup is reflective of the activity of very ancient cut and paste transposable elements, DNA transposons, most of which are, all of which are not active as mobile elements in humans, with the exception of a few elements that have been co-opted, like the RAG recombinase machinery that's responsible for VDJ recombination. About 1% of our DNA, just to put all this in perspective, is exons of unique protein coding genes. And so, again, just sort of underscoring the rich composition of our genetic makeup that's owed to the activity of transposons. So I'll focus on mostly active elements and variants coming from them in humans today. And so a lot of that biology will take us to consider activities of the line retrotransposon. Even though we refer to this part of our genome as the junk genome and sometimes the non-coding genome, lines are actually protein-coding elements that are capable of undergoing a life cycle for their self-propagation that I'll kind of sketch out on this slide. About 20% of your DNA are line elements, about 17% are the so-called line 1 elements, but only a very small subset of those sequences, only about 100 loci, are really still fully intact elements with intact open reading frames and capable of undergoing this life cycle as I'll describe to you. And those loci are largely, but not exclusively, segregating in human populations as structural variants. So we each inherit a complement of potentially active line sequences. If you look at these competent elements, they're full-length sequences, 6 kilobases in size, unlike most of our genome that contains just small fragments of line elements. And they encode 2 open reading frames, named unimaginatively Open Reading Frame 1 and Open Reading Frame 2. The dogma in the field, although I'll admit that it's not so black and white, is that these are kept transcriptionally quiescent in normal cells through methylation of the internal RNA Pol2 promoter sequence, which is a very CG-rich sequence in the first kilobase of the retroelement. But that silencing is imperfect, and we know in lots of diseases, and it's been known for some time in cancers, that there's hypomethylation of line elements, and that can lead to transcription of this bicistronic transcript, a very difficult to detect transcript. That in turn can lead to the translation of the 2 proteins, Open Reading Frame 1 protein and Open Reading Frame 2 protein. The first protein forms a homotrimeric RNA binding complex. We know that it associates with its own RNA. It may associate with other RNAs, that's not sequence specific, in these highly complex RNP particles. It's essential for retrotransposition, but we don't really know what the RNA binding protein is doing. We've got a bit of a better handle on the activities of the second protein, the so-called ORF2 protein, produced in much smaller quantities. ORF2 encompasses an endonuclease domain that's related to the APE endonuclease. Our model in the field now is that nicks our genome at a new, essentially sort of random location with respect to the source element where the RNA and proteins really originated from. And then there's a reverse transcriptase domain that's part of ORF2 as well. And so we think that the nicking action happens first. This model hasn't been completely well worked out. And then you have a cDNA that is extended from the target site, cut site in the genome to copy the line RNA, and then through mechanisms that are yet as poorly understood and may involve host DNA repair factors, you get resolution of that cDNA flap to give you a new double-stranded, ultimately, line insertion in the genome. We know that these line insertions in our DNA are frequently 5' truncated. We don't know if that's because the RT isn't very processive or if there's something that's degrading either the RNA or the cDNA as these lesions are repaired. We do know that this activity is a source of ongoing structural variation in human populations. So there's a basal level of this activity in the germline. We think that an individual in 100 or so inherits, or rather has as part of their constitutional genetic makeup, a line integration site that they did not inherit from their parents. That's really reflective of activity in the germline. And a similar number for SVA insertions. The thinking in the field now is that as frequent as 1 in 20 individuals has an ALU integration. That's a very large, essentially constitutive part of their genome that they didn't inherit from their parents. So if you think about the total of humanity, that means we're testing out tens of millions of de novo line and SVA insertions,
hundreds of millions of de novo ALU insertions, and most of these will never really establish significant allele frequencies in human populations, but some will, we think largely through genetic drift, because most are fairly harmless, and so that's how they end up segregating in human populations. My laboratory at Hopkins was certainly interested in these as structural variants. We know that very rarely insertions of lines or ALUs or SVAs can cripple a gene and cause an outright instance of human disease. That was first actually characterized at Hopkins by Hank Kazazian's laboratory studying patients with hemophilia and recognizing line insertions that took out a coagulation factor gene. My lab at Hopkins then, decades later, was interested in structural variants as potential contributors to human disease with more mild sort of phenotypic impacts. We were interested in common variants segregating in human populations that may contribute to human phenotypes. So I'll just touch on a couple of candidates for that type of activity here with an emphasis on cancer biology. We identified an ALU integration event. ALU are non-coding transposable elements retrotransposed in humans by the activity of line ORF2 protein. And we recognized a specific ALU insertion in an intron of the ARID5B transcription factor. ARID5B is expressed in precursor B cells in the bone marrow, and we got really interested in this ALU because it existed at a location in the genome that had been associated with risk for developing leukemias of that B cell type, precursor B cell acute lymphoblastic leukemias or lymphoid leukemias, which is the most common form of childhood leukemia. So these are, this photomicrograph just shows you those leukemic cells. The leukemia cells and their normal precursors express this important transcriptional regulator. And genome-wide association study groups had recognized that there was a haplotype, a risk haplotype for developing this leukemia, again, segregating in human populations. A common allele conferring a low relative risk, but nevertheless an increased risk very clearly for developing this, this pediatric leukemia. So we wondered if the ALU integration in that intron might be a candidate. We worked with investigators who did the genome-wide association studies to impute the presence or absence, to infer the presence or absence of the ALU insertion variant in a cohort of patients who were part of the genome-wide association study. These are the results of that study. On the x-axis, we have plotted association with disease risk. On the, sorry, that's the y-axis, and on the x-axis here, the position along chromosome 10. So the little dots represent mostly single nucleotide polymorphisms. You can see that there's no particular association as you move towards ARID5B. People who are healthy controls and children who have the leukemia have no difference in sort of their relative inheritance of different haplotypes until you get right to this intron of the gene. And then you can see that the haplotype that people inherit is very strongly associated with disease risk, and that the peak of that disease association is here where the red diamond is. That's representative of the presence of the ALU integration. And then as soon as you move past the ALU insertion to the rest of the gene, including almost all of the coding exons of ARID5B, there's almost no consistent disease association. So in instances like this, we think we have very good genetic evidence that the ALU insertion itself, or a few SNPs on either side of that integration that stay in linkage disequilibrium with the ALU, are really signatures of the risk haplotype. As yet, we've studied this locus. We really don't have a wonderful handle at all on the mechanism for that. We assume that this intronic variant is then affecting the expression levels or perhaps the transcript structure of the ARID5B gene, and that creates a setup, a predisposition for developing this leukemia. But I'm speculating at that point, we really don't have molecular evidence for what the ALU is doing. We have another cancer risk locus that we've been interested in, very similar story from the genetics perspective, but perhaps we've got more of an understanding of what may be happening molecularly. This is an ALU insertion variant, again, a common variant segregating in human populations. on 8q24, which has been associated on a haplotype associated with breast cancer risk. So here's the ALU integration, and we're just showing you in this diagram, which covers a very large genomic interval, that it occurs almost coincidentally with the trait-associated SNPs. If I showed you a sort of higher power, higher resolution view of that locus, it would be very similar to the last ALU story. Here, the ALU insertion variant is in near perfect linkage disequilibrium with breast cancer risk-associated SNPs. And again, the ALU integration is on the risk allele. So women who inherit this ALU integration on AQ24 have a modest but increased risk of developing breast cancer. Probably not significant to inform, more vigilance or screening in any populations. We think that the ALU variant may be having an important effect on gene expression in the interval. The target of this, we expect, is likely the MYC oncogene. That was also the assumption of many of these GWAS groups, because there's known to be looping between these GWAS haplotypes on AQ24 and the MYC oncogene locus, and expression of MYC is really, a very critical contributor to oncogenesis in many tissue types. We know that the ALU variant has enhancer activity. We were able to demonstrate that in ectopic expression assays, and we were also able to do genome editing at the locus, at the AQ24 locus, and pluck out the ALU integration, put back in the ALU integration, and then see an effect on transcript levels of the MYC oncogene in cell models. And so here we have a potential contributor to breast cancer risk through a long distance genetic regulation mechanism. So at this point, I'll pivot away from inherited insertions, and I'll de-emphasize ALU as a type of transposable element and focus on LINE, the protein coding transposon in humans, and its expression and activity in cancers. First, I just want to make a comment about why I think that transposons are being understudied in malignancies or in many different disease contexts or developmental contexts. And that's just that these are very highly repetitive sequences in the genome, and really recognizing that they're differentially expressed in a particular condition is a challenge. And that's especially true of these evolutionarily young protein coding line sequences in humans, which are very highly homologous to one another. And so many people, of course, began explorations of paired tumor normal specimens with an interest in looking at pathways that might have shown upregulation in tumors. And line RNA would never come up in something like that. It's incredibly difficult to detect in malignancies or in any cell type. This in situ hybridization illustrates some nests of ovarian cancer cells, which we know express really high levels of line ORF1 protein, very high degree of hypomethylation of line loci. Here are adjacent normal stromal cells, and the pink is all the line containing RNA sequences. You can see that the malignant cells have tons of line RNA. The normal adjacent stromal cells have tons of line RNA. A lot of the signal is coming from the nuclei. It's part of read-through of gene introns or long non-coding RNAs. And so just doing an RNA study doesn't really reveal the protein coding potential and ultimately DNA damaging mutagenic potential inherent in these retroelements. When we developed an antibody, then, to be able to look at ORF1 protein expression, we could really begin to make progress in the field and characterize what types of human malignancies do in fact express line as a potentially active mobile genetic element. So here I'll just show you, if I can get it to play, a movie that is then the structure of that protein. This was given to me by Oliver Reichenreiter. His laboratory at the University of Tübingen did the crystal structure for the RNA binding protein encoded by line. And very kindly, they marked for me right here at the junction of the unstructured N-terminal domain and the coiled coil domain, the epitope where our antibody is binding. So we have nailed down that sequence at this point, and that's what's being detected here in immunohistochemistry and in subsequent immunohistochemistry slides that I'll show you. Towards the C-terminal domain of the homotrimeric complex is the RNA recognition motif and RNA binding capacity of the sequence. So when we started applying our antibody to human tissues, we saw this very nice dichotomy in many instances where we had normal tissue that expressed relatively little of the ORF1p protein versus adjacent malignancies which tended to express a lot of the ORF1 protein. So in this particular photomicrograph, there's normal colonic epithelium. There is a blush of expression here. We do think that there's some degree of line expression in normal colonic epithelium, but really nothing like the promoter hypomethylation and ORF1 accumulation that we see in the nests of malignant cells that are adjacent. And this is a pattern that we saw again and again as we started to look at different tissues. We saw ORF1 expression in the large majority of breast cancers and ovarian cancers. We initially reported its expression in about half of colon cancers. That probably needs to be revised. Our lab and others are now seeing expression in closer to 80 or 90% of those malignancies. We see pervasive expression as well in lung cancer, also probably higher than our original estimates of about half of those cases, probably closer to 80% plus of those cases. Esophageal cancers, pancreatic cancers. Less studies have been done in prostate cancer to my knowledge, but it also can be expressing line, and there's also good genetic evidence in prostate cancer and all of these tumors for line activity as a retroelement, as I will come to. In some tissues, not in every case, there is this very stark kind of binary pattern of expression where you see almost none or absolutely none in normal tissue, and then an induction coincident with cellular transformation. And probably the best example is in the precursors of high-grade serous ovarian cancer. So we recognized early on that ovarian cancers express a lot of this protein. It actually is the tumor type with the highest levels of ORF1 among the common malignancies that we've looked at. And the thinking from pathologists in this field is that the originating lesions for high-grade serous ovarian cancer occur in the fallopian tube, so-called stick lesions. And so here, for example, is a study that we did in collaboration with Yiming Shi's laboratory at Hopkins, confirmed now by other GYN path groups, shows you histologically normal fallopian tube epithelium where there's no expression of the protein, and then that's in contrast to the stick lesions, the precursors of high-grade serous ovarian cancer, where you can see cytologically the cells look different. They're much more columnar, their nuclei are very open, they're becoming what pathologists will refer to as cytologically transformed atypical cells. In stick lesions, you also see fixation of p53 mutations, and it seems really coincident with that, this very early stage of development of this malignancy, that we see de-repression of the line element and accumulation of the ORF1p protein. So what is the effect then of all of this line expression in human malignancies? At first, I think as a field, we were really thinking about this inspired by Haig's work and work in genetics. We thought that line expression might then create a mutagenic mechanism for compromising tumor suppressor genes in malignancies. So I put up this slide to just give a nod to our recognition now as a field that that does happen. But it is an uncommon contributor to the loss of tumor suppressor genes in cancers. So actually, back in the 90s, there was a group that was looking at the APC locus in colon cancers, and they recognized an instance of a line insertion event that hit a very important protein coding exon in this absolutely vital tumor suppressor gene, and clearly created a mutation that was cancer driving in that individual patient. Scott Devine's laboratory published a beautiful paper in 2016, really illustrating an almost identical type of case from the point of view of the mechanism, and also tracing the line integration to a source element that was active in the individual before any transformation event. And now several groups have converged on the understanding that about 1% of colorectal cancers are caused by integrations of a line element that cripples the APC tumor suppressor gene. So we're seeing this really recurrently. Colon cancers often, almost always even, lose APC but they almost always lose it through other mechanisms. Nevertheless, Lyme can contribute to this type of cancer-driving mutation. That doesn't seem to be what lines most commonly do. When they get activated in cancers, it really seems that we're seeing retrotransposition and lines going everywhere in the genome in a fairly random distribution, although we could come back to some non-randomness about it. And we and others have contributed now to this literature as our tools for line insertion site mapping by next-gen sequencing have really matured. So this particular Circos plot is from a paper that we published on pancreatic cancer. It shows you an instance of a pancreatic ductal adenocarcinoma where we had primary tumor and some distant metastatic sites of disease, and we're taking those different locations, the primary and the met in these two outer wheels of the Circos plot, and marking with hash marks places in the genome where we recognize somatically acquired line insertion sites, where the tumor had a collection of new line integrations that wasn't part of the person's inherited genetic makeup and that we don't see segregating in human populations. And then the centermost wheel marks some of those that are in common between two different sites of disease. So we're even able to do sort of tumor phylogeny studies and recognize a line integration that, for example, happens in the primary tumor and then gets carried to a metastatic site of disease. Now there have been really professionalized efforts as part of the International Genome Consortium to take in the landscape of that retrotransposition, and this just shows you a graph of the overall number of line insertion sites that group is able to sort of pick out in bulk whole genome sequencing, short read sequencing. So not targeted, it's probably not as sensitive as targeted sequencing assays, but still giving you sort of the impression that this, I think, is happening broadly in many types of malignancies, all of which have been cancers where we and others now have recognized their SORF1P protein expression. So the cancer type that seems to dominate is esophageal adenocarcinomas, where you can have hundreds or several hundreds of insertions per sample. It's also commonplace as a mutagenic mechanism in head and neck cancers, in lung cancers, and colorectal adenocarcinomas. Interestingly, ovarian cancers, which I mentioned before, really express a lot of line, are tumors where we don't see as much in the way of new insertions. We frequently only are able to pick out a couple by whole genome sequencing methods, and even with more targeted sequencing approaches, you'll find usually just a handful of canonical, resolved new insertions of lines in genomes. We know only a little bit about the dynamics of the activity of these retro elements in malignancies, but what we know suggests that this is a dynamic process. So Jose Tubio's group, as part of that pan-cancer genome effort and in his prior work with International Cancer Genome Consortium, was able to trace a subset of new line integration events to their source elements. So you could figure out which lines were coming on in cancers. And the way that I think about this is a little bit like that's a moving target, like there may even be a kind of game of whack-a-mole going on such that a line element will become active for a period of time in tumor development and then that activity is lost. So as tumors grow out different subclones, you'll see a whole series of insertions potentially stemming from one source element, which then goes quiet, and then another source element will be active in different branches of the tumor or in specific sort of metastatic sites of disease. And so that activity is something that we really don't yet understand well at all. We used to think that there was just a global de-repression of Lyme. It does seem now more stochastic. We also don't know why a line that had been active might be silenced. It's possible that there are pressures against these. We don't know if it's deleted. We don't know if it's possible for tumors to remethylate and silence these elements. One thing that's clear, though, is that this activity is happening in a broad variety of cancers, that it's associated with protein expression, and then we've got unequivocal genetic evidence for activity of the mobile element. So now we're interested in translational sort of value in that. And one thing that we're beginning to look at, and this is in collaboration with David Walt's laboratory at the Brigham and Women's Hospital, is can we detect that ORF1P protein in the peripheral circulation? So far I've only shown you expression of the protein in tissues, but that's usually at a point where then you have a very clear cancer diagnosis for a patient. So for some period of time, my lab at Hopkins was interested in could we detect ORF1 in the peripheral circulation. We were never really able to do that until we teamed up with David's lab. And David has developed these really incredibly sensitive Simoa assays that are essentially like digital droplet ELISA assays. So they take beads and conjugate a capture antibody to the beads, and then those beads can be segregated into very small micro wells rather than the larger sort of standard way that you would do an ELISA, such that you can come in with a biotinylated detection antibody, and that sort of sandwiching your antigen gives you a fluorescent readout or some other bioindicator readout of your antigen, but you can get a very intense signal because it's not diffused and lost in a larger volume. So it enables detection of proteins at like femtomolar quantities, which is really a much lower limit of detection than is the case in typical ELISA assays. And so the next slide I'll show are unpublished data of our measurements with the Walt lab of circulating ORF1P levels in different types of malignancies. So these are very early days with this biomarker, but I'm sure you'll appreciate my excitement about it. We've done at this point more than 200 healthy controls, and we really don't see this tumor antigen in healthy individuals at all, which is very important. And then as we advance to different cancer cohorts, here's ovarian cancer, here's colorectal cancer, lung cancer, head and neck cancers, prostate cancers, where we were starting to make some improvements on the lower limit of detection of the assay. And we're able to see to varying degrees expression of this protein in the peripheral circulation. So we're very encouraged by these types of data. Now I want to say that these are all patients with very advanced stage malignancies. And you still see that we're missing very large numbers of them. We don't know, but our working hypothesis is now that many of these patients may have an ORF1 expressing tumor and that our blood assays just aren't good enough yet. So we're working hard to develop better antibodies that might bind to the antigen more strongly and further lower the lower limit of detection of these assays with an eye towards use for this type of biomarker in cancer detection. So at this point, I'll switch and talk more about biology, but first I want to stress an important, I think, dichotomy between the two line-encoded proteins in malignancies. So I've shown you that the ORF1P protein is expressed in very high levels in a broad variety of different types of cancers, and that small amounts of that are even leaching out into the peripheral circulation. And I've shown you that that's an indicator of the expression of the line element and its activity as a mobile transposable element, because we can see new line integration sites in cancer genomes. What we can't see in all of this is that second protein, is the protein that must be making these integrations, the ORF2P protein that encodes the endonuclease and the reverse transcriptase. We know it's there. We've got good genetic evidence, right, in the line insertions that we're seeing in cancer genomes, that some of it is there in at least some of these cells, but its expression directly is not something that we're able to detect after spending years developing really great reagents and having a lot of confidence in our antibodies. We think it's expressed at just incredibly low levels, which will be an important point as I get into the next part of my talk, which is on the cellular consequences of line expression, where we're really starting to view this as an important mutagen and a genotoxic, ultimately cytotoxic transposon. So this branch of my laboratory was led by a really phenomenal MD PhD student, Dan Ardelian at Hopkins, who came in and looked at our early data on ORF1 expression in cancers and some of the very early data showing line insertions in malignancies. And he asked what, in retrospect, is a naive but really important question. And that is, does line promote cell growth? We had expression of this retro element in all these different types of cancers. And so Dan just took cell lines that didn't express line at baseline and asked what happens if he creates a doxycycline-inducible construct and turns on this retro element. How do the cells do? How do they like that? And the answer was such a resounding, stark one that that it's really inspired a lot of our subsequent sort of characterizations of line effects on cells. So these are massive overexpression systems where we're producing the biosystronic transcript and measurable amounts of ORF2, unlike human tumors. And cells don't like that at all. So here's some very early experiments with RPE cells where I convince you, I hope that Dan can grow some cells. He expresses GFP and he can still clone cells and they grow and everything is fine. The DOX-inducible system itself isn't doing anything terrible to the cells, but when DOX is driving line, you can see that the expression really is something that stops cell growth. In these particular cells, these are P53 competent RPE cells, and they have just almost no tolerance for line. There's almost no clonogenic growth of the cell line that's possible in the face of line expression. Even in hardier cells and HeLa cells, in 293T cells, and other cells we've worked with for years then since, when we turn on line, the cells don't like it, viability goes way down, although it might not be completely dead. And then if you express a line with mutated reverse transcriptase or endonuclease, you can do a lot to mitigate that cytotoxic effect, really suggesting that it's the ORF2 protein and enzymatic activities intrinsic in that DNA damaging part of the retroelement that is important for this effect. So then Dan wanted to know, does line retrotransposition compromise cells in a context that might be more relevant to human malignancies? And does it create, in that type of context, particular molecular vulnerabilities? So to address that type of question, he took his RPE cells, he knocked down very effectively, it's as good as a knockout, we think, P53, so that the cells could tolerate some degree of line expression. And then he had a TET on line arm of the cells and a luciferase control. He then did a CRISPR knockout of every gene in the genome and grew out these cells in a cell fitness screen, at which point in time over subsequent serial sampling over 27 days, and at the conclusion, Dan was able to recognize guide RNA representation in these cells, reflecting gene mutations ultimately that affect the growth of the line 1 expressing cells. So the way the screen was set up was that he could identify both gene knockouts that were advantageous uniquely in line expressing contexts. Interestingly, APC is one of those genes, and he was also able to recognize a number of synthetic lethal interactors, genes that aren't so important for the growth of RPE cells normally, but that become vitally important in the cells that are coping with retrotransposition. And so those will be the genes that I most focus on now. We recognized from that screen that we got a lot of components of the spliceosome complexes. We think that this may relate to the accumulation of R loops on chromatin in their loss in the absence of these factors, although we haven't really dug into it very much. The next group of genes that came up very highly in the screen were components of the DNA repair pathway, the Fanconi anemia pathway, as well as some replication stress signaling factors. And so that'll be the focus of most of what I describe next. So Dan recognized that really all of the components of the Fanconi anemia DNA repair pathway, best known for its resolution of interstrand crosslinks, become essential genes. That's why they're colored here in red when he turns on line, that there was a synthetic lethal type interaction between loss of this DNA repair pathway and expression of the transposable element. That was true for all of the core components of the Fanconi anemia DNA repair pathway. It was true for FANC-M, which recognizes lesions. It was true for the I and D2 components of the effector complex. And here Dan just did single knockouts of a subset of those genes of FANC-M, A, L, I, and D2, and turned on line and recognized that while those gene knockouts were well tolerated in normal RPE cells, the induction of line and in combination with these knockouts led to a hugely dramatically reduced potential for the cells to be cloned. That, of course, was not true with a non-targeting control guide RNA. He also went on to show that line expression is activating this DNA repair complex. So you can take RPE cells and not do anything to them and see very few FANCD2 nuclear foci as a readout of activation of this pathway, although you see some in S-phase cells. And then if you treat the cells with hydroxyurea, you see many more of these foci. That's a known activator of this pathway. When we express a wild-type line retroelement, we see a similar degree of activation of the Fanconi anemia pathway as read out by the presence of these nuclear foci. And he has other markers of this now published in his paper. When we express a line that is mutated for the RT domain, that seems to have a suppressive effect. So what we're thinking now is that it really is not just the endonuclease cutting DNA, but it's the creation of this intermediate of retrotransposition, this cDNA on chromatin that is so toxic to these cells. At the same time, I just want to make the point that line insertion efficiency is also increased when you compromise the Fanconi anemia pathway. So in parallel, totally independent orthogonal studies from a group at Stanford and a group at NYU showed that if you take out components of the Fanconi repair pathway, you see more line retrotransposition as read out by a GFP reporter of retrotransposition. Also nice evidence that the Fanconi anemia pathway is limiting insertions and limiting the cytotoxic effects of line expression. So now we're also thinking about DNA damage broadly in these cells. This particular set of Western blots is done now by my group at Dana-Farber, building on these findings, where postdoc fellow Carlos Mendez-Durantes is looking at gamma H2AX as a readout of DNA damage. And you can see in the same type of DOX-inducible line expression system, he can turn on line at the usual sort of levels that we have in control cells, and he doesn't generate tons of DNA breaks, double-stranded breaks, really very similar to the uninduced cell control. But when we add lots of doxycycline, we really drive very high line expression with robust levels of WRF2, then you start to see double-stranded breaks under these circumstances. But much less line is more genotoxic, creating many more double-strand breaks in the Fanconi mutant sort of backgrounds. And so further evidence that that pathway may be important in limiting DNA damage caused by line 1, and that DNA damage is a dose-dependent phenomenon. So thinking, again, just broadly about the screen to summarize some of the hits, cells undergoing retrotransposition seem to be particularly reliant on replication-coupled DNA repair. They need Fanconi anemia complex genes. They need, interestingly, BRCA and its partners, BARD1, BAP1. They require replication stress signaling factors, non-essential components of the 911 signaling complex, and the ATR interacting protein. ATR, of course, wouldn't come out on our screen. It's an essential gene. As well as replication fork restart factors like Bloom helicase, Werner helicase, and Werner interacting protein, all critical for the expression, or rather continued growth of line-expressing cells. So now at the Farber, we're interested in whether line and its associated DNA damage might be an important contributor to chromosomal instability that we see in cancers. Up to this point, I think the field has been very focused on line insertion events, and we've assumed that those are what we see in germline events, a line integration that then sort of leaves flanking sequence intact. But we've had some glimmers of evidence from the Pan-Cancer Genome Consortium that line integrations could have broader impacts on chromosomal stability. Some line integration events, for example, occur at breakpoints where there are large deletions on chromosomes. This has been reported before, but was then something being seen again in these whole genome projects. So we turn again to our doxycycline-inducible system. Now, Carlos taking over that system at the Farber, along with bioinformatics fellow in the lab, Jennifer Carlo, and our bioinformatics collaborator, CZ Zhang. And what Carlos and his team are doing now is a very straightforward sort of experiment. He takes p53 null RPE cells. Those are normally like pretty diploid cells with stable genomes. Despite that loss of p53, there's not gross chromosomal instability in these cells. He has a DMSO control and a doxycycline inducible arm. Then he sorts out single cells, and we do whole genome sequencing on the single cells and also grow out clones so that we can begin to ask whether retrotransposition has this sort of broader effect on chromosome stability. And what we're seeing in doing these types of experiments is indeed that line expression can really wreak havoc in these genomes. So here's an example of this whole genome kind of sequencing across chromosome 5 in one of the single cells where he's pulled a cell from the line expressing cohort, and you can see the intact allele is always represented in the shallow sequencing at about 1 copy number, and then the altered allele here is shown in black dots where we have a terminal deletion of that one end of chromosome 5. We do rarely see terminal deletions in the p53 mutant RPE cells alone, but the numbers of these really go through the roof when we express line in the system. We also see, uniquely in the line expressing cells, these duplications of regions, as well as this sort of oscillating pattern that CZ and David Pellman have seen before and associate with chromothripsis. So then when we have some of those types of patterns in single cells, we can do deeper whole genome sequencing and really get at what is occurring at these different breakpoints. And we're able to sort of put together a series of events. In this particular case, CZ has done that, recognizing on 2 different chromosomes a rearrangement that involved probably a spontaneous DNA break on one, and then a line integration of actually a pseudogene at a site of the other, such that you have the very terminal ends of chromosome 15 and 7 fused together with a very clear signature of an ORF2 mediated pseudogene integration event at that place. And then we see tons of rearrangements, which we speculate is then caused by this acentric kind of structure being sequestered in a micronucleus where the nuclear membrane is not intact, and there's a lot of subsequent rearrangements, as you can sort of see in looking at read pairs that are scrambled in their associations there. So over the course of the evolution of a tumor, this sequence presumably would be lost, but it suggests that a line was responsible for one of the chromosome breaks, and so then might be inciting the beginnings of genome instability that could later compound. When we grow out clones in these experiments, we can see how those types of breakage, fusion, bridge cycles evolve, and how this DNA damage can have a compounding effect. So I've shown you up to this point, just single cell cloning events after very short exposures to the line retroelement. But when we grow out progeny clones and sequence later, we can see, for example, not only the terminal deletions illustrated here, but this pattern of adjacent subclonal loss, really suggesting that when you break off the end of a chromosome, it sticks to something else, and when you have instances of dicentric chromosomes, then you can have subsequent breakage with successive mitoses in the cells. So at this point, I'll summarize what I've told you up to this point about line expression in cancer, and just make a few more points before I wrap up. Line is expressed very clearly and active as a mobile genetic element in cancers. I think there's now very strong evidence from lots of groups using many different approaches in the field, and broad consensus on this. We're now getting very interested, although I think it's still a fairly new idea and understudied, in the genotoxic potential of the ORF2p protein. I think very clearly in experimental systems where we can overdrive the protein, it hinders cell growth and creates specific reliances on DNA repair pathways. And so our working model right now is that the line insertion intermediates that are generated by this protein are DNA lesions that pose conflicts for DNA replication, and that line activity may represent an important endogenous source of replication stress and chromosomal instability in cancers. Now, there may be ways to exploit this biology. Right now, as I mentioned, in real malignancies, they're different from our experimental systems because there's much lower levels of the ORF2 protein. We're now really thinking about what are the mechanisms that might regulate the translation of this downstream cistron or other factors that may lead to accumulation of the protein or clearance of the protein with an eye towards targeting those types of mechanisms as a cancer therapeutic. We're still a very long way away from that, but that's kind of our thinking today. Another area that I think is especially interesting in cancer biology, but I won't have time to do more than touch on, is the idea that line expression may be inducing interferon responses, as has been proposed for many types of transposable elements in disease contexts at this point. We see in our RPE cells that line induction really produces very huge quantities. This is a log2 fold change scale of interferon stimulated genes. It's one of the largest gene sets that goes up. We see in these cells, which do not have cGAS-STING, some induction of double-stranded RNA pattern recognition receptors, but we don't really know what to make of that because in many of these systems we're using a recoded line element that shouldn't really have a huge propensity for binding with other line-containing RNAs. And so exactly what the underlying mechanisms are in RPE cells are as yet unclear to us. It is clear that there are consequences for that induction in these cells. So for example, one of our synthetic lethal interactors, where the loss of the gene is very poorly tolerated in line expressing cells, but kind of okay in luciferase controls, is the ADAR gene, which is known to be a type of sensitivity that is induced in cells with innate interferon signaling. So this is a figure that I pull from the science perspective that you just mentioned, just emphasizing that transposable element loci are diverse and that different expression can have different effects, likely in very different cell contexts. Some of this is quite speculative on our part and on others, but these are some of the working models in the field. We're thinking that transposon expression may impact the expression of nearby genes through epigenetic mechanisms, and that could be an important way in which they contribute to cellular phenotypes and maybe disease phenotypes. Specific examples are few at this point in malignancies, but certainly exist, and there are also reports of chimeric transcripts fusions between the transposable element and adjacent genes. We're also getting really interested in just cellular consequences of DNA damage and the potential for innate immune signaling coming from transposable elements. And it's possible that these highly repetitive sequences are generating double-stranded RNAs that could trigger pattern recognition receptors. It's possible that through reverse transcriptases that they encode, they could produce cytoplasmic cDNAs, still controversial and a point of speculation in the field, but that may be an important thing recognized by the same type of innate immune signaling pathways. We're getting really interested in DNA damage that could be caused by line activity and chromosomal instability that we believe this activity is capable to generate, and that may be a source also of cytosolic DNAs or poorly encapsulated micronuclei that trigger innate immune signaling, all of which may be important in how, for example, growing tumors are perceived by the immune system. All right, so with that, I'll stop and try to save a couple minutes for questions. I do want to thank a number of people who've contributed to this series of studies really over several years. I called out Dan Ardelian, who started our self-fitness screens at Hopkins. I'm really excited that he's going to follow me actually to the Dana-Farber, where he'll be a medical oncology fellow soon, although he'll be rightly looking for postdocs in other areas. Nemanja Radic and Marty Taylor were phenomenal trainees in pathology who worked with me at Hopkins, and Marty continues to work with us at the MGH, which is where he is now He was also the mastermind behind our interactions with David Walt's lab. So Nemanja and Marty have worked on ORF1 and ORF2P expression detection in tumors, and all of our ORF1 blood assays are being done with David Walt and a phenomenal postdoc fellow in his lab, Connie Wu, who I co-mentor. I mentioned Carlos and Jennifer along the way as we are taking on our chromosomal instability project with Changzhang Zhang, and Phil Schofield is a technician who's working with us on that project. We've had a number of other great collaborators give us ideas about interactions of line with DNA repair machinery, and our studies on the Fanconi anemia complex in particular have been informed by conversations with Alan and Dipanjan. So with that, I'll stop and take any questions.
Thank you, Kathleen. Fantastic. Any questions? Eric?
Yeah, so that was very cool. Thank you. I have many technical questions. So, so if these transposable elements are essentially a source of additional DNA damage, is that related to BRCA status in breast cancer? And is that something that might potentially be exploitable with something like PARP inhibitor?
So you're asking about BRCA in breast cancers or ovarian cancers for that matter. I don't think we've done enough work to really relate the two, but I think it's a very important area for us to begin to think about more seriously, and for others to join us in thinking about more seriously. So we're thinking right now that BRCA is like the Fanconi anemia complex, required for the growth of at least RPE cells when we massively overexpress line. It's one of our strong synthetic lethal interactors. And so it's certainly possible that line-associated DNA damage has an interaction. I would favor that it would have an interaction with that sort of pathway. At first blush, when we look at stick malignancies or the precursors of ovarian cancer and high-grade serous ovarian cancers, about 90% of those cases express very high levels of ORF1P. About 10% don't. We had the idea that the ones that would not would be the BRCA patients, that for some reason then the coincidence of the loss of BRCA and the induction of line would not be tolerable, but we don't see that that's the case. On initial surveys, it seems that the malignancies from these BRCA patients also do have ORF1. We don't know if that's coming from line elements that have ORF2. We don't know if there are new integration events of line in those malignancies, or if line is a contributor at all. I think it's a really interesting question, but still an area where we don't know a lot. I think where we are as a field with human tumors is that we know there's expression, we know there's retrotransposition ongoing, and in experimental systems, we sort of know that that can contribute to chromosomal instability and create requirements for DNA repair pathways. But understanding whether that genotoxic sort of potential of line is relevant in the malignant contexts, or whether the expression levels are just so low that that it's not a major contributor, I think is something that we don't know broadly among many types of cancers.
So you show that line expression creates huge DNA damage in cycling RPE cells. So I'm wondering if that damage is caused by the line nicking and insertion, or a combination of that with the cells growing. So if you were to do the same experiment in quiescent RPE cells, how much damage would line expression create?
Oh, that's a really interesting point. So I definitely share what I think is an intrinsic assumption in your question, that that continued cell cycles can compound damage. And I think we have pretty clear evidence from that, because when we have a short exposure, which is still enough time for the cells to cycle, admittedly, we can see some events like these terminal deletions. And then when we grow out clones, we can see evidence, although we've only done shallow sequencing at this point, we haven't done the deeper work or single cell sequencing in those samples. We're working on it. But we see preliminary evidence of breakage fusion bridge cycles that then are probably DNA damage that started with line as we're thinking about it, but that line is not required to then, create in successive cell cycles. To your point more specifically though, we have never looked at this in non-cycling cells. It might be a very important experiment to take cells that aren't replicating at all and ask then, do you have insertions at all? That's controversial in the literature of our field, but some groups have reported that you can have canonical insertions. Can you wreak havoc more broadly? I'm not sure the answer to that. I think it's a really interesting and important point.
I have a question about your detection of line 1, ORF1P in ovarian cancers and breast cancers, and primarily those would be female patients. But there's also this hypothesis that men having a Y chromosome and that being so full of repetitive elements that males should have more expression of transposons. Do you see that hypothesis might be contrary, that you might see more line 1 or 1P expression in female tumors versus male tumors? I saw like prostate seemed to be also lower. So
prostate may be lower. I think though some of this relates to just where we are as a field. So the only prostate data that I'm really aware of, and certainly the only data that I showed, is from one of our initial surveys of many different tumor types with an imperfect immunohistochemistry that we didn't want to overinterpret. And so we undercalled really the prevalence of expression in lung and in colon and quite possibly in prostate. We went back in that group and looked more closely. So ovarian popped out just because it's really higher there. We went back and looked more closely at breast cancer, specifically because we also borrowed, as part of our antibody validation, a polyclonal ORF1 antibody from Chris Harris, and he had previously reported expression in breast cancer. And we wanted to, really try to very carefully assess that and match our breast tumor cohort to his. And so we looked a little harder and found higher proportions of cases that were positive in that specific case. Men certainly have esophageal cancer and colon cancer and lung cancer and many tumors where we've seen line expression. And I don't know the proportions of prostate cancer that express, but that could likely be an underestimate. None of these elements that I can think of are coming from the sex chromosomes in the instances where people have identified specific source elements. So while differences related to male-female biology may be at play, I don't think we have a strong indication now that it's just the female cancers or that it has anything to do with an active element on the sex chromosomes.
Okay, cool. Another question, Garrett?
Much more basic. Is there a mechanism for loss of repetitive elements in general from the genome?
Is there a mechanism for their loss from the genome? Sort of. In the case of like ALU sequences, it's very clear that there are tendencies in the germline for non-allelic homologous recombination events that delete intervening sequence, of course, but then also reduce the number of ALUs. For the most part, these line integrations, they have less of a propensity to do that. It can happen, but our thinking in the field is that once an integration goes in, it's a pretty faithful sort of marker of progeny ever after, that there are perhaps very many layers of host mechanisms to prevent this, but then once you get an integration that can be propagated in a population or population of cancer cells, really without any special likelihood that it would be lost. There's no excision pathways that cleanly take out a transposable element integration event.
Okay, if we don't have any further questions, let's thank Dr. Burns for the